In our previous video, we established the idea and structure of an operon that's found only in prokaryotes, and we looked specifically at the lac operon. I want to continue our look at the lac operon, but this time I want to focus on something that's a little bit outside of that realm, in the sense that we're going to be looking at things known as repressor genes. So we're going to entitle this next flowchart Repressor Genes. These are going to be critical genes involved in regulation. Now, one thing that we have to really understand and sort of make sure that's very clear is that repressor genes themselves, and the reason why there's a totally new flowchart for repressor genes is because that they are not a part of the operon, not part of operon. So if we imagine our operon structure that we drew before, you have to imagine that way on somewhere, maybe even some other chromosome, maybe way down the line of that DNA molecule, there's a whole nother section of DNA that's known and codes for a repressor, okay, that has repressor genes on it. Specifically, we can imagine that, let's say we have an operon over here, right? This is our operon, okay? Operon, but somewhere, let's say all the way downstream or upstream, there's a totally separate sequence. Let's do it over here. That's known as the repressor region. Okay, we'll call this repressor genes. These repressor genes are not a part of the operon, thus I drew them separate. They might even be on a separate chromosome. They are not part of the operon, and thus they are going to be necessary in order to code for a repressor. REP in this flowchart will be represented for, or mean repressor, not replication. So code for repressor protein. Now, this repressor protein, so imagine we have repressor genes here. I'm going to draw an RNA polymerase molecule. Let's say it's a triangle. This RNA polymerase will transcribe all of these genes, so we'll do some transcription. There'll be some translation a little bit later on, and then finally we're going to create a protein. And this protein that we're going to create is going to look like this structure that I'm about to draw. Okay, it's a very specific reason I'm drawing it like this. We'll see why. So this is going to be a protein. Specifically, this is a repressor protein. Labeled it as a repressor protein. Now, what we have to understand about repressor proteins is that they are actually considered constitutive in this situation. Okay, so a repressor protein is a constitutive or constitutive, whatever you want to call it, repressor. It's called a constitutive repressor because lactose, okay? Because lactose, we're still talking about the lac operon, so imagine this is lac operon. It's a constitutive repressor because the lactose is not always there. Not always there. So remember how we talked about the situations at which we express lactose metabolism genes? Well, almost all the time we're not going to express lactose metabolism genes because lactose is not always in our E. coli environment. We're not constantly drinking milk, right? So the intestinal environment will not constantly be full of lactose. Thus, it makes sense that this operon, this structure, will repress the genes that make lactose metabolism enzymes until lactose is there. So this is a very key distinction. I think this will make a lot more sense if we sort of elaborate on those situations that we established in our previous flowchart by looking at the presence of lactose and the absence of lactose. So let's look at those situations one more time. Let's imagine we have no lactose. Okay? Let's imagine we didn't drink milk and our intestinal environment, which has E. coli in it, which have lac operons, does not subject itself to lactose. This means that we're going to have an RNA polymerase molecule. Let's imagine an RNA polymerase molecule within this E. coli happens to, let's say, bind to the promoter. Okay, it binds to promoter of, let's say, the lac operon specifically, LO. So there's nothing to stop this RNA polymerase from binding, just simply binding to the promoter. Now, what's going to stop it from binding and then transcribing? That's obviously going to be a repressor. Let's talk about that. A repressor is actually going to be, so if you remember, next to the promoter is what region? An operator region. Specifically, the repressor that we just coded for, this thing right here that was made downstream or upstream somewhere else, not on the operon, this repressor that we coded for, this repressor is actually going to bind to the operator region. Binds to, so it literally floats all the way back to this operon and gets on and attaches itself to the operator region. To operator, let's call it the binding site. The operator has a binding site, and the repressor is a very good fit for this operator's binding site. Okay? So we have an RNA polymerase molecule, and right next to it, we have a repressor protein. Now, what you have to understand is that RNA polymerase has literally and physically been blocked. RNA polymerase is blocked. It's completely blocked because there's a repressor protein right in its face saying that you cannot do gene transcription. So there is no gene transcription, and thus there is no gene expression. Remind yourself what genes should be transcribed and expressed. Those lactose metabolism genes, like lactose permease, like beta-galactosidase, like galactosidase transacetylase. All those things, will they or will they not be expressed if there's no lactose? Of course not. Why not? Because somewhere in this cell, there are genes being expressed because they're constitutive. They're always being expressed because they're going to then translate themselves into this protein that's going to come to the operon and say, hey, there's no lactose, no need to make these, thus you don't need to have these enzymes. I think a good way to understand this is to do a very simple drawing, because it will really sort of drive home this idea of this repressor protein. So let's imagine, I'll do it over here, we have our operon, right? And we have this structure of a promoter region, and then right next to it we have an operator region, and then right next to it we have genes 1, 2, and 3. At this promoter region, what binds here? RNA polymerase binds to promoter region. So let's draw an RNA polymerase molecule just like this. It's going to be as a triangle. You can write inside of it pole, P-O-L for pole. At the operator region, look what's going to come in. This guy right here is going to travel and attach itself right over here at this operator region, and it's going to attach itself much the same way I'm drawing here. This is our repressor protein, okay? It's going to literally and physically block RNA polymerase. RNA polymerase can't move because there's this big fat repressor protein right at the operator region, preventing polymerase from going to 1, 2, and 3 and transcribing it and expressing those genes. Why is this thing here? Why is it not, why isn't it just, why is it there? Why is it bothering polymerase? Because there's no lactose in our environment. What if there is lactose? So let's say there is lactose in the environment. If there is lactose, lactose will very easily enter the cell. So it enters our E. coli cell. We just drank milk. It's going to enter our E. coli cell, and it's actually going to be converted into something. It's converted into something called allolactose. One word, allolactose. It's just an isomer of lactose, just a different form of it. Don't get too caught up on this conversion. Just say that lactose is converted to allolactose. What is the consequence of this? Well, interestingly, allolactose is actually a very good binder to this repressor protein. Allolactose binds to repressors, so REPs, allosteric site it's called, an allosteric site. So what we have here is the following situation. Still have another operon, right? So I'm going to draw, this is no lactose, so no lactose. We have this blockage happening because of this fat repressor protein right here. What if we do have lactose? Let's imagine there is lactose. So we have a promoter. Right next to our promoter is going to be a nice RNA polymerase molecule denoted by this triangle, so POL for pole. And right next to it is going to be an operator region. And right next to that is going to be 1, 2, and 3 genes. At this operator region, we have this, usually, this protein that's blocking RNA polymerase from going. But now I'm telling you that the allolactose binds to the repressor's allosteric site. What is its allosteric site? I did this on purpose. I drew it like this on purpose because let's imagine I have this nice allosteric molecule, this allolactose molecule denoted by A. It's going to come in and fit real nicely into this perfect circle sort of place that it can just insert itself into. What happens once it binds? Well, this actually is very, very cool. The repressor actually changes shape. It changes shape, it actually, because of the shape change, becomes inactivated. And once it's inactivated, it can't bind to what? It can't bind to this operator region. Can't bind to the operator. It literally falls off. This whole idea right here is known as allosteric regulation. Regulation that is due to shape change. Allolactose is here because lactose is present. Lactose is converted to allolactose to bind to this binding site of our repressor protein that we drew over here. This repressor protein will fall off, so let's eliminate it. Let's imagine it's all gone. Now polymerase is free to go down the line and transcribe 1, 2, and 3. So thus we can state that the genes themselves are TXNED transcribed, thus the genes will be expressed. Again, why are they being expressed in this situation? Because there is lactose. So it makes sense to express the genes necessary to metabolize lactose. Lactose is in the environment. Thus, we're going to regulate the genes that usually say no lactose genes, and tell them to turn on, tell them to be expressed, tell them to be transcribed into those 1, 2, and 3 genes that we already have mentioned. So overall, what we can state is that allolactose in this situation can be considered what? Go back to your terms. Allolactose is actually considered the inducer of this situation because as an inducer, it induces or causes the coding of Operon genes. How does it do this? It does this by physically, allosterically inactivating the constitutive repressor protein that's usually there. So overall, let's imagine that most of the time, this cell, this E. coli, does not have lactose in its environment. It makes no sense for it to constantly be producing lactose enzyme genes, okay? So what is it going to do? All the time, it's going to make this repressor protein out of the repressor genes somewhere far away from the operon, bring this repressor protein all the way to the operon and say, stick it right on the operator. This prevents polymerase from going anywhere because it's physically blocked from transcribing. What if we do have lactose? Lactose comes in, turns into allolactose, fits very nicely right over here into this region. This causes a complete shape change, inactivates the repressor, polymerase goes down the line, quickly transcribes, eventually translates the genes into the expressed mode to metabolize lactose. That is the whole idea of repression and repressor genes in our gene regulation story.